Hello, students. Welcome back to our lecture series on animal cell culture technology. This is Dr. Joyjyoti Das, Assistant Professor of Biotechnology at Adamas University. So let me continue with our series of lectures. And in our last lecture, we began with a discussion on uh, the animal cell culture and what we mean by animal cell culture. So we ended with this particular schematic showing the various forms of animal cell culture. And we spoke about the various definitions of uh, a primary culture, secondary culture, subculture, and we ended with the discussion that when you are culturing cells from healthy normal tissue, then those cells will stay for a definite number of divisions in your artificial culture. But then if you are taking cells from cancerous tissue, those cells will stay maintained in culture indefinitely since they do not have a finite lifespan. Okay, so with those definitions in mind, let us continue our discussion. And now, in this particular lecture, what we are going to do is we are going to understand why you would do culturing of primary cells, normal primary cells, or say culturing of cancerous cells taken from a tumorous tissue, and how they are important in the medical sector or health sector. What are the various applications? Okay, so let us begin discussing slightly the differences again between a primary and continuous cell culture. So let us look at this table. So in case of primary cell culture, your source will be a mixture of cells derived from the tissue of origin. So, and that tissue of origin can be from a laboratory animal, like that of a, uh, I mean, an experimental mice, a lab rat, or any other test organism, or it can be directly from a human through a human biopsy sample. And in case of continuous cell culture, the source of the continuous cell culture would be a mixture of cells derived from, again, human or animal uh, tumors. Okay, and as we have already talked about this, tumors are cancerous tissues, and cancerous tissues have infinite lifespan. Okay, so in the next point on, you can see that primary cell cultures or cell lines derived from primary culture will have a limited lifespan. They will usually divide only a limited number of times before losing their ability to proliferate. And this event, as I mentioned in the previous lecture, is called as senescence or aging of a culture, whereas continuous cell culture will be uh, subcultured indefinitely. And morphological changes, yes, you can note that also to distinguish primary cell culture from continuous cell culture. In primary cell culture, all the cells will retain their normal karyotype, that is the genetic constitution, that the number of chromosomes, the size of chromosomes, everything will be similar as that of the tissue of origin. But continuous cell lines will not display the same karyotype. Cancer cells, as we know, have high mitotic rates. They have high genomic instability, which is why their chromosomes, there will be changes in their genome, in the number of chromosomes, or in their the size of chromosomes. And so they will not display the same karyotype as that of the parent tissue. So given these two types of cell culture, we are going to now emphasize that which can be used and for what application in cell culture, which is important at what stage of your experiment based on the basic premise of the experiment that you have set in cell culture towards a specific application. So first we need to understand now the what are the various purposes of uh, knowing or learning animal cell culture methodologies. Okay, so directly let me take you to the application areas where cell culture technology is currently playing a major role. So number one is cell systems act as very good model systems for studying basic biology, interactions between disease-causing agents and cells, effects of drugs on cells, process and triggering of aging and nutritional studies. Okay, so what does this mean? Say you have designed a drug for something. Okay, you have designed an anti-cancer drug. Okay, and you are claiming that it has the ability to kill cancer cells because that's what cancer scientists are trying to do. They're trying to understand uh, how cancer spreads, and they're trying to target one of the pathways that cancer cells are taking help of to grow and proliferate uncontrollably. And they're targeting that pathway or any signaling molecule of that pathway or any receptor on the cancer cell, and they're trying to like contain the cancer. They're trying to regress the tumor and stop it from spreading, right? So you have synthesized, suppose you have synthesized a molecule, a new molecule, a novel molecule that has not been reported before to exhibit anti-cancer property, and you want to uh, study its anti-cancer properties. Of course, you won't get a friend or a stranger of your same species, that is Homo sapiens, to volunteer you uh, in your experiment. Of course not, because there will be uh, several ethical concerns. There will be, several inhibitions to be your model system. And so we won't find any human being to test your drug on at the first instance. Plus, the law of the land will prohibit you from doing so. So, what can you test your drug on? So, you have to test it on uh, model systems. Okay. So, you will say, I'll take a lab rat, I'll take a mice. Okay. But then it is also an organism, it is also a mammal, and there will be ethical concerns if you want to sacrifice animals at will for establishing your experiment or your proving your theories. So you have to first find more simpler model systems on which you can test your drug, on which you can test whether the drug is at all exerting its desired property, the property that you projected it to exhibit. Okay, so those model systems are cells. Okay, so as I said in the previous slides, that you have two types of model system in hand. One are your primary cells, and the other are your continuous cells or the immortalized cells. So again, for isolating primary cells, say you are you have designed a drug for liver cancer, okay, to treat liver cancer. So you need liver cancer cells. And you also need to ascertain that the other healthy cells of your liver that are around the tumor should not get affected by it, and neither should the other uh, healthy primary cells of your body be affected by that drug. Otherwise, that drug does not stand as an anti-cancer drug. Anti-cancer, I mean, liver-specific anti-cancer drug. It does not qualify for being called so, right? So for that, you would require to test your drug on liver cells, primary liver cells taken from a healthy tissue, and as well as the liver cancer cells taken from a cancerous tissue. Okay, which of these is easier to find? Of course, the uh, liver cells taken from cancerous tissue, because once they have been isolated from a patient, they will be there in culture, they will be maintained in culture, and uh, actually there are several repositories in our world which maintain these cells, which keep these cells in the form of a library. And if you write to them and send them a subscription, they will send you a flask of those cells. You can purchase them and you can perform your experiment on those cells. Okay, so cancer cells are obviously easy to find since they're immortal and they can be continuously maintained in culture. So one such repository for cell is the National Center for Cell Sciences located in Pune in India. Okay, so once you bring those uh, cancer cells to a laboratory, you can grow them, you can culture them, you can maintain them in culture, and you can take some cells and apply your liver drug on them, I mean, anti-cancer drug on them, and see whether they are able to kill those cancerous liver cells. What effect is the drug exerting on the normal cancer cells? That also needs to be evaluated. And for that, you can purchase some primary cell lines that are also being maintained in the uh, uh, cell repositories. And but that's the second step of the experiment. So primary cells, will stay for a finite lifetime in culture. So first you will test on liver cancer cells, because they, once you purchase them, you can maintain them in the laboratory. And in future slides, I'll teach you a technique how to preserve those cells as well in your lab when you are not using those cells. So as you used to make glycerol stock of bacteria for preserving your microorganisms and use them when you would require to use them, you can also similarly preserve animal cells in your laboratory using a certain technique called as cryopreservation, and you can again bring them to life in culture when you need them and test on them. So of course, your first step will be to use a cancerous liver cell as your model system. Once it is successful, once you see that you're getting some anti-cancer activity on those cells using your design drug, you will move ahead to the next step, wherein you will buy the primary liver cells and you will test your drug on them to analyze toxicity. So if your drug exerts toxicity on them as well, on the healthy cells, healthy liver cells as well, then you are going to disqualify the drug. So because it is not cancer specific, it is not only killing the cancer cells, but also the healthy cells, which nullifies your hypothesis, right? Okay, so that is how cells can be used as model systems for developing a drug. Okay. The second is toxicity testing. Study the effect of new drugs. Okay. So, I mean, similar to the first point, you need to like analyze how much toxic a drug is to the cells of your body, right? To say maybe how much toxic is a drug, is an antibiotic drug, anti-diabetic drug, for example, that you have proposed to the liver cells of your body, right? So for testing that, you need to take primary liver cells, and you apply your anti-diabetic drug on them and see what changes it is uh, exerting in those liver cells, because you are taking a drug to contain your diabetes, but in the process you're affecting your liver. That is not desired. That is not at all desirable. Okay. So for toxicity testing of drug, your uh, cells will again come in handy. Okay. Third is cancer research, which is again a similar point, because I gave you the example of cancer only while talking about model systems. So, in cancer research, you can use cells to study the function of various chemicals, viruses, and radiations to convert normal cultured cells to cancerous cells. So, basically, to study the process of carcinogenesis. Carcinogenesis means what? Is the process of development of cancer. Okay, and we know that cancer occurs due to mutations in the genome. Okay, and where does the genome lie? Genome lies inside the nucleus, inside the cells, right? And nucleus is inside the cells. So again, you need a model cell system on which you can study the effect of a carcinogen. Carcinogen is a substance that causes cancer. Okay, so how is it causing cancer? What, through what pathway it is causing cancer, or where in the genome it is mutating the DNA? All that you can find out. All those scientific explanations you can find out, provided you have a model cell to study it, right? Next, in virology, cultivation of virus for vaccine production, also to study their infectious cycle. Okay, so we know viruses have host specificity, and viruses can grow in bacteria cells, they can grow in plant cells, and they can grow in animal cells as well. So for those viruses which grow in specific animal cells only, you can uh, bring those cells in your laboratory from the cell repositories, and you can study by injecting that particular cell with the particular virus, and you can see what are the phenotypic changes. You can study the protein expression levels, and you can figure out the pathway through which the changes are taking place. And you can also cultivate the virus in host cell, because viruses will always need a host to multiply. And for making vaccines, right? What are vaccines? Vaccines are attenuated uh, organisms or parts of organisms, microorganisms, that you inject into a body of a host, and you like do what? You wait for your immune system to develop antibodies against that particular organism or part or organism, so that you can fight the organism uh, in the future, right? You can fight the organism using your own immunity. Okay, so to produce vaccines, of course, you will need to grow the virus in large numbers so that you can make a lot of vaccine against them. Okay, so not against them, by using them rather, because these vaccines will be injected into the patient, and the patient eventually will develop antibodies against the a particular virus. So to cultivate that virus, you will require host cells. Okay. Then comes genetic engineering. This is the production of commercial proteins, large scale production of viruses for use in vaccine production. Again, So in vaccine production, you'll be requiring to like again use viruses and engineer the viruses or engineer the fragments of say membrane proteins of viruses into suitable hosts and grow them in large numbers. And also you can extract some proteins which are nothing but parts of those viruses, and you can use them as vaccine components. Okay. So all that comes under the field of genetic engineering. And there is another very important field which directly deals with medical interventions nowadays in different types of uh, uh, diseases, because it deals with manipulation of the DNA, which is central of all life forms. And we all know that the information is stored in genes that are contained in DNA within the chromosome. So, say a cell that has a functional gene can be replaced with cells which are having non-functional gene, or vice versa. Okay, so cells having a functional gene can be replaced to cells which are having non-functional gene. So a cell which is having faulty gene, say for example a faulty gene, can be and due to which the person or individual is having a particular disease, can be uh, corrected. The condition of that person can be corrected by replacing that faulty gene with a correct copy, with a copy of the correct gene inserted into its genome. And that comes under the field of gene therapy. Okay, for that also you require the culturing of cells, because in often in gene therapy, the ex vivo gene therapy is done, wherein cells from the tissue, defective tissue of a patient are taken out into a petri dish or into culture vessel. They're cultured, and the faulty part of the genome of those cells are modified with an exogenous insert carrying the correct gene, and that correct gene is then transferred into the host genome by replacing the faulty gene, and the cells are then put back into the patient. The corrected cells are then put back into the patient. Okay, so all that comes under the purview of gene therapy. Okay, so to sum up this part, let us see in this particular schematic, which shows the various applications of tissue culture. So if we were to classify tissue culture into like basic and applied sciences. So if we were to like classify the applications into basic sciences and applied sciences, what are the various applications that would come under each of these heads? So in basic sciences, means you are exploring science, you are trying to generate scientific knowledge, you're trying to create a wealth of knowledge in the domain of cellular science. So in that case, you can uh, do what type of studies? You can do studies of intracellular activities such as DNA transcription, protein synthesis, energy metabolism, drug metabolism, cell cycle. So all these are taking place where? Inside the cells. So for that, you again need cells to study them. You can study intracellular flux, such as RNA processing, hormone receptors, metabolite flux, signal transduction, of course, because you can you know about techniques like Western blotting, by which you can study the entire set of proteins being synthesized in the cell at a particular point of time. You can analyze the differential gene expression by studying the mRNA through techniques such as uh, uh, reverse transcriptase, PCR. Okay, and you can do a whole lot of genomic and proteomics analysis, such as Like you can study cell phenotypes and metabolic pathways. Okay, you can also study cell-cell interactions because you will grow cells together in a plate, cells sitting side by side. What is the exchange of chemicals taking place, and uh, like uh, what is the paracrine control of one cell over the other? How cell-cell interactions are affecting cell proliferation, metabolic cooperation, adhesion, motility, or matrix interaction and invasion properties of cells. All these can be studied under uh, the basic applications of cell culture technology. And what is the applied part? We have already talked about toxicology, wherein you will analyze the toxicity of a new drug. You will analyze uh, infection of a microorganism into a particular cell. What is the pathogenesis? You will study uh, immunology in such cellular systems. You can study the cytokines and inflammation and other immunological signaling pathways, and you can also do like produce vaccines in cells that we studies that we already studied in the previous slide. You can study the action of drugs under pharmacology, ligand receptor interactions, drug metabolism and resistance. And there is a separate field called tissue engineering, wherein you can also repair a fractured bone by actually making a construct of uh, cells and extracellular matrix that is essentially called a tissue construct. And that tissue construct, if you graft it into the defect of your bone or say skin or any other wound, that tissue will help the cells of the surrounding uh, organ to grow into it and eventually become a part of your body. Okay, so all these are the various applications of cell culture. And in this next slide, or cell or tissue culture, I would say. And in this next slide, I'm going to again summarize the different forms of tissue culture to you. We already have discussed this in definition format. Here you can see a schematic of it. So you can either grow. So all these are commonly placed under the umbrella term tissue culture, but there are different forms such as organ culture, wherein you're culturing the tissue in its three-dimensional form as it would occur in vitro. And there is the explant culture where you are culturing part of the tissue as an explant that you have taken out from a particular organism, and then the cells will start migrating out from the tissue into the culture vessel, and the cells will start growing independently. And the other form of culture is this or dissociated cell culture, where you are taking a tissue from a patient or a healthy individual, and you are mincing the tissue. Not mincing, I would say you are dispersing the cells of the tissue into a single cell level, and then you are using that disaggregated tissue cell form the monolayer at solid liquid interface. Means you are dispersing the cells and then you are growing them on the surface of your culture vessel. And in organotypic culture, you are actually creating a 3D environment wherein you are trying to achieve a tissue-like growth, wherein different types of cells will be co-cultured with or without matrix. So matrix is an attachment for cells, and we are going to talk about it in future lectures. And what you're essentially doing through this is you're co-culturing different cells as they would occur in a tissue by re-aggregating them from the single cell level. So this is what you mean by organotypic culture. So this table somewhat summarizes the or compares rather the different forms of culture. For example, in culture, it will be from adult tissue fragments or embryonic organs. Explant culture will be again from tissue fragments. Cell culture will be from disaggregated tissue, and organotypic culture may be from component primary culture or cell lines. If you compare the effort, then in organ culture, the effort is much higher. In explant culture, it is moderate. Cell culture is having the least effort involved in it, while organotypic culture involves moderate effect. Okay, so characterization, in terms of characterization of organ culture, it is easy by histology. This age-old lab technique, you can actually view the internal components of the organ. In explant also, using several markers and cell-based studies, you can uh, uh, characterize the explant. And for cell culture, you can do biochemical, molecular, immunological, cytological assays. So this is also very convenient. And organotypic culture, you would require to perform histology, confocal microscopy, or magnetic resonance imaging. So histology will be again informative, difficult, not applicable and informative. And in case of quantitation in organ culture will be difficult. Explant, again, quantitation of cells will be difficult, but it's easiest if you're doing single cell culture. In organotypic culture, it may require image analysis. So what are the various advantages of cell culture? The advantages we have already talked about. You can control the pH, temperature, osmolality, and the dissolved gas concentration, because you're culturing the cells in vitro, so you can vary the parameters as you want, and you can create different artificial parameters under which you can study the uh, genetic and phenotypic effect of the cells. You can vary the microenvironment of the cells. You can give it different parameters. You can vary the parameters. You can regulate the matrix composition. You can change the cell-cell interaction by growing different types of cells in different geometries. And uh, availability of selective media, cell cloning, these are certain advantages in cases of cell line when it comes to cell line homogeneity, and characterization can be easily performed. So you can easily study the cells if you're culturing them at a single cell level, or DNA profiling, immunostaining. These techniques are there to further characterize your cells, and these are very convenient techniques. Okay, what is of another very important aspect is that the scale, that is the number of replicates, can be increased substantially. You can grow a lot of cells together to like at a particular instance of time to like perform your experiments on different sets of cells and different batches of cells to like establish your particular experiment. And so it will be also time saving. So as the time will be reduced, and reduction of animal use. This is one of the most important uh, things to keep in mind when it comes to cell culture. As I said at the outset, that if you're using a cell culture for establishing a drug, what you're essentially doing is you are reducing the sacrifice of animals, and of course bypassing the like experimentation on humans, which you will not be allowed to do in any way due to the law of the land. Okay, but then there are certain limitations of tissue culture also. For example, this requires a lot of techniques which can be performed by skilled labor only. So, and one of the most important aspect is sterile handling of the various instruments and tools that you use in animal cell culture. avoidance of chemical contamination, detection of microbial contamination, and quantity and cost. So this is cost intensive. The animal cell culture is cost intensive, and it requires a lot of expensive reagents if taken together. Okay, and identification of cell type may be a limitation, because in histologic type of culture, histotypic culture or organotypic culture, uh, it is difficult to analyze the uh, uh, different characterization of the composition cells, and it may not always be like easy to recapitulate or recreate the uh, original tissue while you are culturing in vitro in whatever close resemblance possible. And there might be heterogeneities in genetic and variabilities in the cell culture due to genetic instability. If you're culturing them, if you're culturing a set of cells for a long time, there might be phenotypic instability due to like random mutations, dedifferentiation, adaptation, selective overgrowth. So all these things will might create a problem in your experimental data set, and which is why there are certain procedures to follow and a lot to learn if you're stepping in to do animal cell culture. So uh, I'll end this lecture here and see you again soon with another interesting lecture on our lecture series, Animal Cell Culture Technology. Bye.